Define electrochemical gradient and how it affects the movement of charged particles across the membrane. As we've talked about previously, ion concentrations vary on either side of the membrane, and the fact that there are varied concentrations of specific ions across the membrane is going to contribute to chemical disequilibrium. We don't have an equal concentration of sodium inside of the cell and outside of the cell. We don't have an equal concentration of potassium inside of the cell and outside of the cell. And so what we're going to see is that we're going to have more of these blue kind of diamond-like structures. These are going to be sodium, so we've got more concentration of sodium outside of the cell than inside of the cell. That is going to generate a concentration gradient for sodium. If we take a look at chlorine, chlorine are the little green dots here. We've got more chlorine, a higher concentration of chlorine outside of the cell than inside of the cell. And so that is going to generate a chemical force, concentration gradient, trying to move the materials, chlorine, from outside of the cell inside of the cell. The flip side of that is that we've got a higher concentration of potassium, these little purple kind of squares. We've got a higher concentration of potassium inside of the cell versus outside of the cell. So in this example, sodium, chloride, and potassium are all in a chemical disequilibrium state. They have different concentrations inside of the cell and outside of the cell. And so that is going to contribute to the chemical portion of the electrochemical gradient. And so we're going to have driving force of the concentration gradient. In addition to that, if we take a look at the overall charges that are going to be present, we are going to have more positively charged ions, more cations outside of the cell than we're going to have inside of the cell. And so what this means is that the inside of the cell is going to be more negative than the outside of the cell. It's going to have more negative ions, known as anions, and so this is going to generate an electrical force. And so this is illustrated here. We got little minus signs that are indicating the negative charge inside of the cell. We've got the little pluses here indicating the positive charges outside of the cell. Okay, so if we take a look at something like sodium, we said were going to be these blue diamonds. These have a concentration gradient that is going to try to push these ions from outside of the cell inside of the cell. It also is going to have an electrical gradient. Sodium, with its positive charge, is going to be repelled from the positive charge on the surface of the membrane. And so it is going to be drawn electrically, unlike charges are attracted to one another. These sodium ions are going to be attracted to the negative charge inside of the cell. In contrast, our chlorine over here, chlorine has a chemical gradient where we have a higher concentration outside of the cell versus inside. So we're going to have a chemical force moving it inside of the cell, but then chlorine has a negative charge. And negative charges are going to be repelled by the negative charge inside of the cell, and they're going to be attracted to the positive charges here. So in the case of chlorine, we have a situation where the chemical force is moving in one direction, the electrical force is moving in the opposite direction. And so what we can see with these membranes, with this electrochemical gradient, is that if we can selectively open up the membrane, open up ion channels to selectively allow one specific type of ion to move through it, those ions can move through it, change their concentration gradient, change their electrical kind of distribution, and we could result in a change in the electrical charge across the membrane. And that's going to be important when we start talking about both neurons and muscle cells. So to emphasize this a little bit further, that ion movement is controlled by both its chemical concentration and its voltage gradient, so that distribution of the charges. And so on the example here, we're taking a look at potassium ions that are located at a higher concentration inside of the cell. And so we've got a concentration gradient that is going to try to move our potassium ions down its concentration gradient if we've got a potassium ion channel that's opened up. But then we're going to have a voltage gradient because these positively charged potassium ions are not going to be kind of attracted contribute to the positive charge it is accumulated outside of our cell. So in this case, with potassium, we've got a concentration gradient pushing them outside of the cell. We've got a voltage gradient or electrical gradient pushing it back inside of the cell. And again, depending upon the specific concentration and the strength of the voltage will determine if we open up an ion channel, which direction these ions are going to be flowing. So to further emphasize this, we can see that movement of ions across the membrane can cause a change in the membrane potential, basically cause a change in the ion distribution and concentration of positive charges and negative charges across the membrane, which is going to contribute to electrical changes, electrical measurable changes, voltages across that membrane, which contributes to the term membrane potential. Now, a cell at rest, so we're looking at like a nerve cell here, is going to have our potassium ion channels open up a little bit. And so we're going to get a little bit of leakage of potassium ions outside of the cell down their concentration gradient. And that's going to maintain that negative charge within the cell itself, a positive charge outside of the cell. If we've got depolarization, which we'll talk about with our nervous system, we're going to trigger a change in the ion channel properties. We're going to trigger a change in the permeability of the membrane to sodium, calcium, and potassium in the example we've got here on this slide. We are going to close our potassium ion channel, so potassium is no longer able to flow across the membrane, but we're going to open up our sodium channels. We're going to open up our calcium channels. Both of these are going to be driven by their electrochemical gradient inside of the cell. So sodium is going to be pushed inside of the cell. Calcium with two positive charges is going to be pushed into the cell. So that what happens then is that these positive charges are going to accumulate inside of the cell, and we're going to make the inside of the cell now more positive than was it was at rest. And that's going to be important when we take a look at the properties of nerves and muscles.